Our first speaker is Michael Hiller from the Max Planck Institute of Molecular Cell Biology and Genetics in Germany, in Dresden, and he will speak about identifying genomic changes that are associated with or cause phenotypic changes between species.
Thank you very much for the introduction, and also thank you for the invitation to this great workshop. It's really a pleasure to be here. So I guess this is probably a strong title because I put this this C word here in it for a reason, and in a way I would like to in a way show you how we try to associate phenotypic differences between species to differences in their genomes, which is something we can now do given that many species have sequenced genomes, and would like to discuss in a way view when and under which conditions we can predict that maybe this is a causal change that we found. And of course, I'm happy to any feedback that you guys would like to give me. Okay, so for me, one of the most fascinating aspects of biology is this great phenotypic diversity that evolution has produced, right? And you can see here species that can dive, or mammals that can dive for more than 1 hour. We have flying mammals, we have birds that can hover on the same spot. We have creatures with body armor, mammals that have lost eyes, or reptiles that lost limbs, etc. And I think that this diversity is probably what Charles Darwin meant when he concluded The Origin of Species, writing that endless forms, most beautiful and most wonderful, have been and are being evolved. And in the genomic era, all the species shown here, and of course many others, have sequenced genomes. And this allows us now to address what I think is a fundamental question. And that is, what is the, I guess many of you cannot really see this, so what's the genomic basis that underlies these phenotypic differences between species? And this is kind of the main question that my lab tries to address.
Conceptually, when we think of genomic changes, it makes sense to put them into different categories. So one category could be changes in regulatory elements that would then lead to maybe a different expression of the gene, or leading to more or less expression of the gene. There are changes in the protein coding sequence which can affect or tune the function of the protein, or can lead to a novel function. Novel genes arise in evolution by duplication, or also de novo from previously non-coding sequence. And ancestral genes can also get lost in evolution. And then, of course, there are many other types of changes, right? I show you those 4 because this is what we put some work in the past. But in this talk, I will only focus on those 2 types. So talking about differences in regulatory elements in the first part, and talking about losing genes in the second part. So specifically, what I'm going to do in the remaining kind of 30 minutes, I would like to show you and also explain kind of the methods that we're using to associate phenotypic to genomic differences at a very basic level. Then I will address, or in a way show you, that we think sometimes losing genes is actually important for adaptation. This may be a strong claim. And at the end, I guess I'd like to discuss a bit association versus causality, and what's potentially needed to distinguish the 2, and where are definitely what I see are limits of what's feasible. Okay, the first part, to associate phenotypic differences to difference in the genomes, I would like to stress we're not talking here about individuals or populations of one and the same species, which is something that much research worldwide focuses on. We are focusing on different diverged species. So we developed a framework that we call forward genomics, in analogy to forward genetics, because we are starting with an ancestral phenotype that, as you can see in this phylogeny here, was changed or lost in independent lineages. And the idea is then we can run a genome-wide screen, that the genome of all these species is here, this line, right? So imagine you could stretch out the ACGT letters that are in each of those genomes, And then we can ask for every region that is overall conserved among these species here, we can ask which of those are conserved or present in precisely those species where this trait is present, and which of them have sequence divergence or are maybe completely lost in those species where the trait has been changed or is lost. Right? This is how we try to associate a phenotypic difference pattern to the divergence or loss signature of a particular genomic region. And so here's how we do this, how we detect such associations. This is a kind of a phylogenetic tree, and if we now focus on a single conserved element, which for the most part is something like a few hundred nucleotides, right? And this is something like how the nucleotide alignment could look like. These are the sequences at the tips here, and we can use maximum likelihood in this phylogeny to infer how these ancestral states, so those internal nodes, looked like. And this allows us then to compare the start and end of every branch and measure how much of the sequence is still or has been conserved on a particular branch, right? So if you now focus on a particular branch, we can then simply count how many bases in this alignment are identical, and in this toy example we have 11 out of 15 bases that are identical, so we'd say 73% of that sequence is conserved on that particular branch. And then we can classify those branches into two groups, into the green ones here. Along those we think that the trait was preserved, and the red ones we think that the trait was lost. And this you can either use parsimony or also likelihood approaches to infer this. So now we have those two groups of branches, and for each branch we have a value that measures how much of the sequence of this particular genomic region is conserved. And what's important here is that the sequences at the tips, they are of course related, right? Because there's a phylogeny behind how these sequences evolved. But these per branch conservation values are independent, because if I dissolve a tree into its branches, this by definition represents independent evolution, right? For the most part, if you have no introgression or hybridization or something like this. So this is an assumption we make. And then we can ask if the trait loss branches typically have lower conservation values than the trait preserving branches. And we've used simulations and found that Pearson correlation has the highest power to distinguish that. And so we can now, in a way, ask if there's a significant Pearson correlation coefficient. And if so, then we would say we found an association between this phenotypic pattern here and the sequence divergence of this particular genomic region. And then in a way we can march over the entire genome and look at thousands of conserved elements and in a way rank them by their p-value, do FDR correction, etc. So let's get into a specific example. So if you look at this phylogeny of mammals, you see that there are four independent creatures or lineages that adapted to a subterranean lifestyle. And this is always associated with eye degeneration. And in those two species here, and I'm not sure you can see this one, it's probably too black, those two species in a way dig tunnels with their forehead. And this is likely the reason why the rudimentary eyes on those species are actually buried behind skin and fur, so they are truly blind. So we were interested in what are the genomic changes that are associated with eye degeneration in these subterranean mammals. So forward genomics conceptually looks at conserved genomic regions, and they largely fall into two classes. In the first class are exons of coding genes, and here we find preferential sequence divergence in these subterranean mammals here, in exons belonging to a total of 208 genes. And then of course the question is, well, which of those 208 genes could be relevant for eye degeneration? right? And I say this because those mammals, of course, probably share also many other traits in addition to having degenerated eyes. Living in these burrows means you have to deal with hypoxia, for example, right? There's low oxygen content. And there are probably a whole suite of adaptations that was necessary to thrive in this environment. And in a way, we only have 4 lineages, right? So to figure out which of those 208 genes are relevant for eye degeneration, which in a way was the motivation for this study, we can look at what is known about the molecular function of those genes, right? And for then I say on purpose molecular function, right? Not evolutionary function. And of course, when you do this, you always, if you give me a set of genes, right, the chance that you find a few eye-related genes that have known eye-related functions is always there, right? So we were asking whether those 208 genes are actually statistically enriched in eye-related functions. And this was the case. So you see here, on the y-axis, the minus log 10 of the adjusted p-value, so everything behind this dashed line here is significant after false discovery or after correcting for multiple testing. And you see that, I just show you a few of those Go terms like visual perception, lens development, and structural lens components like this comprises many crystalline genes, for example. That these functions, molecular functions, are statistically enriched. And this was also supported by other studies. And then in mammals, when we compare mammalian genomes, the larger part of these conserved genomic regions are sit in non-exonic, so they don't overlap exons, and many of these non-exonic elements, or CNEs, they overlap cis-regulatory elements, so those that drive expression of genes in different time points and tissues. So here we screened a set of 350,000 of these elements, and we found significant sequence divergence in these subterranean mammals in about 9,300 of them. And then, of course, we asked the same question, right? Which of those could be relevant for eye degeneration, and is there any enrichment? And this is a more difficult question to ask because, in contrast to the genes, we typically don't know much about what these non-coding regions are doing. So we've been using, in a way, 2 statistical approaches to assess this. And the first approach assumes that many of these CNEs have regulatory activity and that they may regulate either the upstream or the downstream gene. So we can associate each of these elements with potential target genes and then ask if these associated genes are enriched in certain molecular functions. And we found that these diverged CNEs tend to be located in the vicinity of genes that have eye and in particular lens-related functions. But this is kind of indirect evidence. Stronger evidence would be to use functional genomics to ask if these diverged CNEs overlap eye regulatory elements. And the idea here is that we can use approaches like ChIP-seq or TAC-seq or something else to in a way get a species that has normal eye development, and we get developing eye tissue, perform this experiment, and therefore get genome-wide a set of regulatory elements that are active during normal eye development. And then we can overlap these diverged CNEs with this functional data, and we can use the non-diverged CNEs as a control, and in a way then run a Fisher's exact test to ask if we have any preferential overlap. And then we can repeat the same exercise with regulatory elements from other tissues to ask if this overlap is specific for eye-related regulatory activity. So we've been using a method that's called a TAC-seq, which is a very fast and powerful method to profile regions in the genome where the chromatin is largely open, and this is typically a hallmark of regulatory activity. And we've been using mouse in a way as a, well, we have a mouse house in the institute, so we can, in a way, get tissue of this creature. And we've extracted embryonic eye, and for comparison also embryonic limb and midbrain tissue. And when we then do this overlap between the diverged CNEs and our I-ataxic data, we see that there is a significantly higher overlap than what you have for the non-diverged CNEs. And when we look at our limb or midbrain data, or also publicly available data of like skeletal tissue, then we don't see any preferential overlap. So this tells us that this enrichment is, or that the diverged CNEs are enriched in genomic regions that are I-regulatory elements in mouse. And this enrichment appears to be rather specific for I-related regulatory activity. So to sum up this part, with this forward genomics method, we found that there are, well, several hundred diverged CNEs that are located near I-related genes and that overlap I-regulatory elements. And together with the results for genes, which is something that we and also other labs have worked on, this implicates that both genes and also or divergence in regulatory elements is likely involved in eye degeneration in these species.
How far can a CNE be from a gene before you no longer think it's affiliated with that gene, the exons of that gene?
This is in a way purely arbitrary what we're doing right now. We assume that we go to each side with a maximum of 1 megabase. And the 1 megabase comes from the fact that I think the sonic hedgehog limb enhancer, which is located, that's located roughly a megabase away in mouse. And this was for a long time kind of like the longest distance that was known. I think by now there are some enhancers for IRX developmental transcription factors which are located even further away. This is a bit arbitrary. Maybe what's important here is that what we are of course missing are those associations when the enhancer actually jumps over a gene in between, right? And that appears to be quite frequent. And statistically, if you assume each of those enhancer regulates either the upstream or the downstream gene, in the best case, we're making one false association for every CNE, right? But the whole point here is that all these false associations, they will just increase noise, and if we still detect a significant enrichment for genes having a particular molecular function, then this is probably good evidence that there's some sort of preferential location near genes of a particular class. Okay, in the second part, I would like to talk about losing genes in evolution. And the motivation for this project in a way came from the observation that this forward genomics method that I explained to you just measures plain sequence divergence. So we just count how many nucleotides differ, right? And it's a very crude measure. Of course, it's nice because it gives us an unbiased approach to look at all the different conserved elements in the genome, regardless of what they do, right? The downside is that we ignore knowledge about the different classes of elements and which mutations are likely to affect their function, right? And if we think of coding genes, we know pretty well which mutations inactivate a coding gene, shown here by, this is a coding exon, alignment between two species with the splice sites here, right? Of course, one possibility is you can delete the entire thing. That's kind of obvious. But in mammals, this appears to happen not so frequently because genomes are quite large and there is no real pressure to have compact genomes. So there is a lot of, I guess, junk DNA, right? So what's much more frequent are smaller mutations that shift the protein reading frame, right? So afterwards you get a completely different protein coding sequence, and often you run into a stop codon. Mutations that destroy splice sites, which typically lead to not including this exon in the final messenger RNA. And of course, base changes that create an in-frame stop codon, right? So our goal was to systematically identify these mutations across many mammals, and we had to do so at a very high accuracy. And if you look at this toy example here, this probably appears like a straightforward problem, right? Because given such an alignment, I mean, we can all do this by hand. And this is in a way the when we started, this is what we thought it's going to be easy. But then it turned out that if you want to do this accurately across many, many genomes, that is actually quite challenging. And we encountered a large number of issues that have to be addressed. And I'll not talk about all of them, but I'd like to give you kind of a flavor of where the predominant issues actually are located. In this, the shifted splice site is one frequent issue. So here you see an alignment between, this is real data, between human and cow. This is the 3' splice site, which has to be an AG. And you see here in the cow, there's a G to C mutation, which should destroy the splice site and therefore should lead to exon skipping. Well, the cow is annotated, and we can simply look up what is annotated for the cow, and it turns out the splice site has shifted 9 bases upstream. So effectively, this just makes the exon 9 bases or 3 amino acids longer, right? So no evidence for gene inactivation. In this case, we have a 4-base insertion in the cow. And if you carefully look at this alignment, it's quite similar, right? This looks like really a decent, there's no reason to suspect this alignment is wrong. But it turns out you can find an alternative alignment, and this is not an alternative fact, this thing really exists, okay? Where pretty much the same 4-base insertion is now located in the intron. And the reason is, if you carefully look, there is some sort of micro duplication here. So the aligner doesn't really necessarily know where to put this insertion. And if you put it in the exon, you would conclude, oh, this looks like a gene loss. If you put it in the intron, we would conclude nothing happened. So there were lots of, it took us kind of 3 years to address these issues here as best as we can, develop new methods to do so. And I'll not show you those details, but if we test this final pipeline now, because in a way we address several steps in a row, and we test it on a large set of genes that are truly conserved between, I think, human, mouse, rat, cow, and dog, then the pipeline achieves a specificity of, so meaning not calling a conserved gene as lost, of at least 99.7%. So we think it's quite accurate, and it in a way makes few mistakes on a genome-wide level. And then we applied this approach to a large genome alignment that included human as the reference, and then in total 62, sorry, some things are cropped here, 62 other placental mammals, and the way we generated catalogs of lost genes. And this allowed us then to run this forward genomics method, not looking now at nucleotide divergence, but now we can focus only on the genes, and we have this information whether a gene is intact or lost. And so now we can associate lost or changed phenotypes directly to gene losses. And we hope this would increase power, right, to see relevant changes. So I'll give you, I think, 4 different stories of where we applied this kind of principle. And the first one was, the first trait was looking at dietary specialization. So we grouped mammals into strict carnivores and strict herbivores, and then we screened the data to find genes preferentially lost in herbivores and genes preferentially lost in carnivores. In the data set we had, we excluded all omnivores, which are somewhere in the middle if you polarize herbi and carnivores. And we had, if you look at these species here, we had 6 independent herbivore lineages, and we had 5 independent carnivore lineages. And we found 37 genes preferentially lost in herbivores and 44 genes preferentially lost in these carnivores. And of course, again, the question is which of those could be relevant for dietary specialization. And for example, when we look at molecular function and kind of all the annotations we have of genes, I think the top gene associated with or preferential loss in herbivores had something to do with oocyte activation, so reproduction. Clearly nothing, so potentially a trait that is shared among the species, but definitely not related to diet. So nevertheless, looking that when we look at molecular function and when we try to interpret what those genes do as best as we can, we found 2 genes which are likely relevant for diet, preferentially lost in herbivores and 4 preferentially lost in carnivores. And when you think of losing a gene, there are, maybe roughly speaking, 2 main principles that could explain why a gene got lost in evolution. Use it or lose it, and less is more. And I would like to explain both of these principles using a gene that is lost in this group and a gene that's lost in this group. So we found that a gene called NR1I3 is lost in 3 independent carnivore lineages: whales and dolphins, in insectivorous bats, and this Starner's mole here, which is eating earthworms and creatures like this. This gene encodes a receptor that activates a whole suite of xenobiotic metabolizing enzymes. So those are enzymes you need to detoxify and metabolize foreign compounds, such as those that are contained in plants. So we think that this gene was likely lost as a consequence of having a lower exposure of plant-derived xenobiotics in these very strict carnivores. So the hypothesis of why this gene was lost is in a way use it or lose it. Those species had no need to maintain the gene because having it or not having it didn't provide the selective advantage or disadvantage. The other gene I'd like to talk about is called PNLIP-RP1, and this is lost in 8 independent herbivorous lineages, such as those shown here. And the function of this gene relates to the digestion of dietary fat, so triglycerides. To digest triglycerides, you need the pancreatic triglyceride lipase. And this enzyme is only active if it is bound to another protein which acts as a cofactor. PNLIP-RP1 itself has no enzymatic activity, but it can bind and sequester this cofactor. And that means if you have this gene, then you inhibit the triglyceride lipase by competing for binding with the cofactor. And this effect you can actually see in mouse knockout studies where this gene is selectively inactivated. And they could measure that enzymatic activity is drastically increased. And those mice on a regular diet pretty much double their fat content. So inactivating this gene provides a mean to improve the efficiency of digesting dietary triglycerides. And given that these plant-based diets, what these guys eat, are very low in triglycerides, which otherwise is a great source of energy, we think that is a gene that could have been beneficial to lose if you want to adapt to this particular diet. So this could be a case of less is more, where losing ancestral genes may have been beneficial for these species.
Did you mean that there are 8 independent gene loss events, or do these all trace back to a founder species?
No, these are really independent. So we can look at which lineages, we can look at shared inactivating mutations. So sometimes you see between sister species that exactly the same stop codon occurs. And this you can trace back to an internal branch in the tree, telling you this is likely the branch where the gene was lost. And so looking at the herbivores in our data set, we think it's lost at least 8 times independently.
And what about the omnivores? If you go back to the whole lineage tree and look back at all the species you have?
This we haven't done, but I can tell you this gene is actually lost in 2 carnivores. The sperm whale and baleen whales. And this is very exciting because the diet of sperm whales are large squid, and the diet of baleen whales is to a large extent krill. And both squid and krill also contain very low triglyceride levels. So we think it could have, so in a way this is an exception when you group species as a very crude measure into carnivores and herbivores, right? And we have, again, with insectivorous species, carnivorous species. So we all group them into carnivores. But of course, the actual composition of the diet can still vary. So this, I think, is evidence that would further support that it could be beneficial to lose this built-in break, so to say. Another interesting trait we looked at was having body armor. And you see here that the Chinese pangolin and the armadillo down here are the only two mammalian lineages that have body armor in the forms of scales or scutes. And we found that a gene DDB2 is only lost in those two species compared to all the other mammals we had in our data set. DDB2 is an enzyme that is required to detect DNA lesions which are caused by, or DNA damage caused by UV light. And then it triggers repair of these lesions. And if you inactivate this gene, where there are some human individuals which don't have a functional version of this gene, this is associated with xeroderma pigmentosum, which is hypersensitivity to sunlight. So those are people who can, in a way, never go outside when it's daytime. They can only play, those kids can only play at night. And a similar effect was shown in knockout mice, where these mice are highly susceptible to developing UV light-induced skin cancer. So we interpret the loss of this gene as that potentially the scales and the body armor of these species, which in a way covers pretty much the entire dorsal surface, protects those species from sunlight well enough that they are potentially able to lose the gene without having any deleterious consequences. So we think this is another case of use it or lose it, because obviously losing that gene is probably not causally involved in creating body armor, right? There was a question in the back.
But can you tell if they lost the genes first or they didn't get armor?
We cannot really tell, because so we can. There are now several pangolin genomes, and we can. So we know in pangolins the gene, I forgot the exact age, but it's not a very recent loss. So this, at least in other pangolins that have been sequenced, they share the same mutations. In armadillos, we estimate that the loss is quite early in the branch. The problem is that I think it's not really clear when body armor evolved, because of we haven't found the right fossils at the right age. So we can't really. Timing is a very good argument. We've been using this for other gene losses in a way to predict whether we have more a use it or lose it case, or whether the gene loss happened at a time when certain adaptations also happened. Well, it coincided with a period during which adaptations evolved. But here we lack the data on the way to tell this. And I would like to show in my last example is we can look for convergent gene losses in aquatic mammals, represented by whales and dolphins, and the manatee, which is a completely independent lineage. And among those genes which are exclusively lost, as far as we can tell based on the genomes available, in these creatures is MMP12. And MMP12 is an elastine-degrading protease that is expressed by macrophages, or certain types of immune cells. And so this gene is linked to a disease called chronic obstructive pulmonary disease. And this is where we got a clue of what this could mean, or why this gene could have been lost in aquatic mammals. So here's a logical chain of arguments. This disease is related to smoking. This is the large risk factor for it. Smoking leads to the recruitment or activates macrophages in the lung. Those macrophages secrete MMP12. MMP12 degrades elastine fibers in the lung, and this in turn leads to a reduced elasticity of the alveoli. And this is why these patients, in a way, have problems emptying their lung, because you need the lung, in a way, to recoil and keep the airways open to actually get the air out of the lung. So hyperactivity of MMP12 gives you an incomplete emptying of the lung. Okay? So you would assume that if we now lose the gene, we should get some sort of the opposite phenotype. And this is precisely what characterizes these mammals here. They have a feature called explosive exhalation, which enables them to empty their entire lung, nearly 90% of the air that's in the lung, in really a fraction of the time. So let me, and this is something you guys have all, oops, that I guess everybody has seen this OSD. Right? When a dolphin comes to the surface, very quickly they can exhale and inhale. And you can try it yourself. It takes us several seconds to exhale, and then probably still 60% of the air actually remain in the lung. These species are much more efficient, and this process is, this I find truly fascinating. Sorry, I can't see the point. This is a blue whale here, which has a 1,500 liter lung volume. And it can exhale and inhale in less than 2 seconds. So what you need for explosive exhalation, and that was shown, is extensive elastic tissue in the lung. So it's possible that losing an elastin-degrading enzyme contributed to the extensive elastic lung tissue that you need for explosive exhalation. To sum up this part, we have a highly accurate method to detect gene losses in the alignments of many species. We think most of the genes fall into this use it or lose it category, so kind of neutral losses. We think sometimes in evolution it could be beneficial to lose ancestral genes, and then gene loss can potentially contribute to adaptations. In the third part, I will quickly talk about association versus causality, because what I've shown you for pretty much all we do is associating phenotypic differences to genomic differences. But what we would like to actually understand when we think of adaptations and traits, which genomic changes are actually cause phenotypic change. And here it's interesting to maybe be reminded of what at least the Wikipedia definition of causality is, and that's something is also partially new for me. It says that causality is efficacy by which one process or state, a cause, contributes to the production of another state and effect. So the cause is partially responsible for the effect, or in other words, a process can have many causes. And I think this describes, if you think of a phenotype pretty well, because most phenotypes, you need many genomic changes to give you this phenotypic change or adaptation, right? So to infer such a link, we of course need to experimentally test whether recapitulating the evolutionary genomic change in a suitable system affects this trait in a direction that we see in the wild. And there are many ways of how you can do this. Protein function can sometimes be tested in vitro. You can manipulate cells in a dish if you have a trait that you can assay on the cellular level. And of course here technologies like CRISPR-Cas9 are very helpful to actually introduce the respective genomic change. You can work on model organisms, but there we're very limited, right? For mammals it's largely mouse, and for then maybe have chicken and fish. What I think could be helpful in future is getting organoids, which now would also allow you to, in a way, experiment with more complex systems of creatures that otherwise would not be amenable to experimentation, like chimpanzees or humans. And so, fortunately for us, there were lots of studies where people did equivalent perturbations in mouse. I showed you that this gene, PNLIP-RP1, has been knocked out, which is essentially what happened in many herbivores, right? And effects were measured. The same for MMP12, right? And people could show that this greatly diminished elastin-degrading activity. So knockout study is very helpful for us to interpret the potential effects of losing genes. And this is an example where, in a way, we took one of these diverged regulatory elements in the blind mole rat, and we used CRISPR to replace the mouse enhancer, or the mouse regulatory element, with the blind mole rat version. So precisely recapitulating on a local context what happened in the blind mole rat. To our surprise, this element is apparently not an enhancer, but a silencer, because now we get embryonic overexpression of this gene. and we failed to see any recognizable lens phenotype so far. So probably telling us this is a change that if it had a very subtle effect or had no effect on this trait in this lineage. Good. What is still not clear, last slide, is whether the genomic change that we found really contributed to the trait change in evolution. Right? So if you think of, here's an example, the evolution of whales and dolphins. So where we know, okay, relevant changes happened on this branch. And you can use mouse to test causality. And you would probably argue, well, in principle, we should maybe test species which are closer to the study objects. So doing a transgenic hippo, that would be hard. Transgenic cow, I think, is to some extent feasible, although you need lots of money and time to do this. Nevertheless, you can never be, I think, certain that if you can recapitulate an effect in these living species nowadays, that this is really was still a causal effect in the evolution of these aquatic mammals. So effectively what we would like to do, and I think this is nearly impossible, we would like to take this ancestral system or organism and introduce the same change that happened in this ancestor and then ask if you can change the trait. Right? I think this getting there is potentially impossible, right? But it's interesting to think about. Okay, I'd like to thank the people who did. I highlight here all the people whose work I talked about. Collaborators, different sources of funding, and the community for, in a way, generating all the data that made this all this stuff possible. Thank you very much.
Can you show us a picture of your people? I want to see whether they're beautiful.
They are. You can't even
They
ask
are.
that question.
I can.
Thank you very much, Michael. Very beautiful people. Question?
Yes, can I ask you two questions? One, a sort of background question. In the confounding issues, in setting up your algorithm, how did you cope with pseudogenes, which are quite common? Are they annotated out in all the species, or is it
So
sometimes
I like to use the term lost genes, because that implies that nothing of the gene or similar copy or version is left. People, we have thousands of processed pseudogenes, which is a non-functional copy of a gene that still exists.
Or sometimes it's a local duplication.
Yeah, so we don't look at processed pseudogenes, and part of the issues we had to deal with is to not confuse aligning a processed pseudogene when the real ortholog is potentially missing in a genome assembly, which at the time we started doing this was still a major issue. So we look at genes where, in a way, where there's no functional copy, as far as we can tell, of this ortholog left in a genome.
I mean, there are examples
Just
of
a comment, they are
pseudogenes
called
being
unitary.
being reused, aren't they?
We like to, I know they're called, some people call them unitary pseudogenes. I think inactivated genes, lost genes is a...
Well, I invented the term pseudogene, and it was a 1977 paper, but as you say, it's
Did I answer
types.
your question, or did I misunderstand it?
I'm sorry?
Did I misunderstood your question about the pseudogenes?
No, I was just asking how you cope with it in your algorithms. Then my other question was, you discovered some very interesting examples, and you give us examples of how loss of genes gives you information about triglyceride synthesis. Is this loss of gene mainly a regulatory loss? Because it's difficult from an evolutionary point to think of just completely deleting a gene. By loss of gene, you mean loss of gene function, do you?
No, when I say gene loss, we find inactivating mutations such as stop codons, frame shifts,
You have
splice site mutations in the coding exons. For the most part, as I said, the remnants of those genes are still present in the genomes. So we can, for example, look if the lost gene occurs in a context of a conserved gene order. And really the gene is lost to an extent that we think it's nearly impossible to still generate a functional protein from whatever is left there.
Yes, right, thank you. Because I mean, you implied that in one of your last examples, it was a regulatory event.
Yes, this is going back in a way to the results from the first part of the talk where we specifically looked at gene regulation. Sorry for that.
Oh, we have so many questions. So I wanted to allow only one, but okay, two.
I was wondering just how much you're relying on current genome annotation and how much you're actually actively looking for the lost genes. Because many genes are available in genomes that are not annotated. And many of these genomes have a tremendous amount of holes. So the fact that genes are missing may not actually be missing in the genome.
Yeah, this is a very relevant point. And the Vertebrate Genome Project group, they want to actually, I think they're writing a paper now that about false gene losses, where genes were not present in the assembly, but they are present in the real genome of a species. We try to avoid this mistake. So the second box in this long list of issues was assembly gaps. So if there's a region that maps to a part of the genome where we know there's a hole in the assembly, as you said, then we are not claiming this is a loss.
I'm not just talking about that. I'm also talking about the actual annotation of the genome.
So we're not using all we use is in a way the human gene annotation. As you need a reference to know where exons are in the human genome. We have a human reference genome alignment. So there's 62, or now we have 120 mammals are all aligned to the human genome. And now I can ask whether a particular exon, where is it conserved and where is it lost?
If you're dealing with genome alignments, though, you may lose copies of the genes that are not in alignment.
That is correct. So right now, one of the few issues we have is if there are gene duplications, then these reference genome alignments like multi-Z, they would only take the best copy that aligns. We are developing a kind of a genus pipeline version 2.0. Well, actually, we are developing a method that uses genome alignments to directly infer orthology. And this is based on those so-called alignment chains. So there we see all duplications. We see similar paralogs aligning. We see processed pseudogenes aligning. We can tell from the pattern of how, not the exons, but how introns and flanking intergenic regions align. We can, with very high precision, can distinguish orthologs from processed pseudogenes and paralogs. And this we can detect co-orthologs, so duplicated, lineage-specific duplicated genes. And so we're now in a way developing a method that integrates annotating or determining orthology and annotating orthologs, determining lost genes, where you would also like to annotate the remnants of those genes in the other genome. And in a way also detecting gene duplications, in a way using, and this is would deal with this duplication.
One other continuation essentially of genome annotation in terms of the splice sites. In the species for which there's a lot of RNA available, do you use it to annotate the splice sites? Because the AC that you had is actually the recognition site of the minor spliceosome, not the major spliceosome. ATAC is also a recognized splice junction.
But this is not a U12 intron. So we have a list of 400 something.
Those are known.
Human annotated U12 introns.
Okay.
What for the large part happens is that U2 introns convert into U12, but the other way around doesn't really happen. So we ignore mutations in splice sites in annotated U12 introns, because they are the dinucleotide diversities. It's ATAC, it's GTAG, and there's a huge variety of other dinucleotides that in certain contexts work.
So you don't use RNA at all to help annotate?
We don't use RNA at all.
Okay.
This is purely on a reference, human reference point of view.
Okay, thank you very much. We'll continue this discussion in the break.
Thank you.